Bismillahirrahmanirrahim. The title of our speech is Green Synthesis of Nickel Oxide Nanoparticles using Artemisia Leaves Extract and Improving Their Antioxidant Activities. The aim of the study: biosynthesize nickel oxide nanoparticles using Artemisia Leaves Extract. Study the effect of nickel nitrate concentration on the size and antioxidant activity of nickel oxide nanoparticles. Abstract: in this study, eco-friendly nickel oxide nanoparticles were biosynthesized using Artemisia Leaves Extract. Then 4 concentrations of nickel nitrate were performed in order to study the effect of these different nickel nitrate concentrations on the size and antioxidant activity of nickel oxide nanoparticles. The prepared were characterized with a diversity of analyses such as ultraviolet visible, FTIR, XR, DSM, and EDX. Finally, their antioxidant activity was tested on following 3 different techniques such as FRAP, CAT, DPPA. Material and methods: biosynthesis of nickel oxide nanoparticles. Leaves of Artemisia were collected in March 2020 from Tbilisi and were washed then dried. After that, the dry leaves were powdered. 10 grams of the powder was mixed with 100 milliliters of distilled water to obtain the extract, and stirred for 24 hours. Then the mixture was filtered to obtain nickel oxide nanoparticles. 50 milliliters of the extract was mixed with different molarities of nickel nitrate at 75 degrees Celsius for an hour. Nickel oxide nanoparticles that formed flow by centrifugation and several washes then dried to make a powder. The obtained was calcinated at 500 degrees Celsius for 2 hours. Characterization of nickel oxide nanoparticles. The biosynthesized nickel oxide nanoparticles were characterized with a diversity of analyses such as ultraviolet visible spectroscopy, FTIR, XR, DSM analysis. Antioxidant activity. DPPH given in assay. Different concentrations were mixed with 2 milliliters of DPPH. The mixture was dispersed in a dark at room temperature, and readings were taken at 517 nanometers against blank. The anti-radical activity was measured using the inhibition percent curve of each sample. Total antioxidant activity. Type test was done using phosphomolybdenum methods. 1 milligram of each sample was applied to 2 milliliters of the reactant. The mixture was dispersed and incubated at 95 degrees Celsius for 90 minutes, and readings were taken at 695 nanometers against blank. Ferric reducing antioxidant power assay. This was performed using Bunsen strain methods. 1 milligram of each sample was applied to 2 milliliters of the reactant. The mixture was dispersed for an hour at 37 degrees Celsius, and readings were taken at 593 nanometers against blank. Results and discussions. Characterization. Ultraviolet visible spectroscopy analysis. Figure 1 displays ultraviolet visible spectra of nickel oxide nanoparticles formed at different molar concentrations. As can be seen, these spectra show only one maximal absorption peak apparent at 307 nanometers. Since nickel nanoparticles have a characteristic absorbance peak in the region of 230 nanometers to 400 nanometers, and these peaks observed agree well with other work. Thus, ultraviolet visible absorption spectrum confirms the formation of nickel oxide nanoparticles. FTIR spectroscopy analysis. Figure 2 shows the infrared spectrum of Busan-sized nickel oxide nanoparticles at different molarities in comparison with the X-type. As shown, some bands in spectrum, the broad band at 3303 cm -1 is attributed to the OH stretching vibration mode. Another weak band at 2345 centimeters -1 showed that there is a triple band of CC or CN stretching vibration mode. Also, the weak band at 1641 centimeters -1 indicate the stretching vibration of CC of aromatic compounds or CO stretching of carboxylic acid. For the spectrum of nickel oxide nanoparticles showed one band at 507 centimeters -1 is due the nickel oxide stretching vibration mode. The FTIR spectra matched the XRD results precisely without any impurities. X-ray diffraction. The XRD pattern shown in figure 3, as can be seen in this figure, nickel oxide nanoparticles peaks were extremely intense and sharp, indicated that the resultant products are highly crystalline. The diffraction peak at 2 theta are well matched with this GCPDS card. The average crystallite size of nickel oxide nanoparticles were calculated using the Debye-Scherrer formula. Table 1 displays the average size of nickel oxide nanoparticles percent size at different nickel nitrate concentrations. Scanning electron microscopy. The surface morphological features of Busan-sized nickel oxide nanoparticles were studied by SEM. Figure 4 shows the same image of the different samples related to the concentrations, respectively. The result indicates that nickel oxides are in spherical shape with intensity in agglomeration. Moreover, EDX analysis of the Busan-sized nickel oxide nanoparticles shown in figure 5 confirms the presence of nickel and oxygen. Antioxidant activity. According to the result, nickel oxide have a significant antioxidant activity. Thanks for your attention.